Okay. So last day we talked about how we can design a particular drug and how to find the key properties and key characteristics of that particular drug. And now, but the main aim is that whenever you find a drug, the drug must bind with something, right? To show its activity, it must bind to a proper target, because without binding to a target, it can't show its therapeutic activity. Now, this is where the target classification is needed, because just a minute. Yeah. This is why we need to classify the drug target, because there are multiple targets in your body. You have protein target, you have DNA target, you have RNA target. Now, among this, the most well established targets are obviously the proteins. It's because the proteins are our worker force, right? So what happens is that suppose anything that happens in our body, it happens due to the dysregulation of a particular protein, either due to its upregulation or due to its downregulation. Now, this upregulation and downregulation is obviously controlled by the DNA, because that's what controls the whole transcription. So if your transcription rate is elevated, and obviously you're going to get it is going to get translated into the during translation, and it's going to be give you a higher number of proteins. And the similar thing happens when you have a very underexpressed mRNA. But it's really difficult to target nucleic acid due to the reason that mostly, in most of the cases, the DNA assumes a double helix structure, right? And the double helix structure, so it's really difficult to find a unique region so that your protein is going to bind to only your desired promoter, not to any other promoters, because the promoter structure is mostly same in every cases. But there are some moieties that are present in the promoters of some oncogenes that helps you to design a target against them, and these are like the I motifs. These moieties are the I motifs or the G quadruplexes or the G triplexes. So whenever you have these moieties, it's better to target them because they have a typical topology, and you can exploit that topology to design a molecule that is that can efficiently bind to that particular target. Okay. But most of the time we have is that we have this protein, and to tell about the so now what is biological drug target? Actually, biological target is actually anything within our living organism to which any other entity like a endogenous ligand or a drug that can bind, and this binding results in a change in its behavior or function. Now, for example, the common classes of biological targets are generally proteins and nucleic acid. Now, this definition is actually context dependent, and it can actually refer to the biological target of a pharmacologically active drug compound. And example, the receptor target of it can also be a receptor target of a hormone, for example, insulin, or there are some other targets of an external stimulus, right? But generally, biological targets are most commonly proteins such as these enzymes, ion channels, and receptors. But why receptors? Why receptors? Can you tell me why receptor is preferred as a target?
Because they bind to protein very easily.
Yes, that's a very good observation. But apart from it, there is another reason, and the reason is that every signaling pathway is actually controlled by the receptor. Yes, it's controlled by the receptor. So whenever you have, suppose as I'm telling you, suppose you have oncogenesis or someone is suffering from a particular case. Now, it is usually due to the misregulation of a particular signaling pathway, like there is AKT pathway, there is MAP kinase pathway, right? Now, all these pathway, they all originate from a particular receptor. So if I can shut down the receptor, or if I can regulate the receptor activity, then obviously the overall whole downstream function also gets regulated accordingly. And while targeting the receptor, the drug that you have, it doesn't have to cross the cell membrane, because there are some ligands that fails to cross the cell membrane. You have to be extra cautious to design a ligand that can cross the cell membrane. It's really easy to design a molecule that fails to cross the cell membrane, and there are several servers that also allows you to determine whether that particular molecule can cross the cell membrane or not, right? So it's really easy to find a target or design a molecule that fails to cross the cell membrane. And if that's so, then obviously targeting the receptor is the best method, because while targeting the receptor, your molecule doesn't have to cross the cell membrane. But if it is a protein targeting drug or a nucleic acid targeting drug, or a protein that is present inside the cytoplasm, then your molecule has to cross cell membrane. And if it is a nucleic acid targeting drug, then the molecule has to cross two membrane. One is the cell membrane, other is the nuclear membrane. So designing a molecular ligand targeting to nucleic acid is a little bit more tricky, and it is really rather easy to target a receptor or to a protein. Understand? And most commonly, the most widely used targets are the GPCR targeted drug, that is G protein coupled receptor, because they actually control almost a major part of biological pathway, the GPCRs. Apart from it, you also have the ion channels and the new ion channels that are present on the surface and that regulate the cell homeostasis. Okay, so these are the major drug targeting system or that you have, that one has. Now, for example, there are some molecules. You can obviously divide this according to several. As I've said, you have to classify these molecules. So there are various kind of classification. One is the small. The smalls, they generally target the enzyme, receptor, transcription factor, ion channels, transport proteins, and all. Okay, and then you have the other biologics. The biologics, the peptide, the proteins, these are the part of biologics, right? Whenever you suppose this for this COVID-19, we are using antibody, right? We are using antibody to we are not treating the antibody, but we are giving in a protein that gives rise to in some form of antibody in our body. And this is also a drug actually. This also is actually a drug, but that falls under the biologics. Okay, that falls under the biologics. So whenever you design a protein or a peptide or some aptamers, you know about aptamers, right? Aptamers are generally the RNA, small stretches of RNA or peptide coupled with DNA. These are the general aptamers, and whenever you have those also falls under the biologics category. Okay, and finally you have a nucleic acid drug, and those are generally the RNAi, that RNA interference. That what they do is simply they move and they bind to the mRNA and they degrade the mRNA and they stop they bind to, they silence the mRNA. And if your mRNA gets silenced, then obviously it's not going to translate into proteins. And so in one way you are actually limiting the production of proteins also. But this only occurs if that particular drug or that particular disease happens due to the overexpression of a protein. If a particular disease happens due to the underexpression of a protein, then of course you can't go through this, because suppressing the mRNA is going to further reduce the concentration of the mRNA in your concentration of the protein and obviously the mRNA in your body. Okay. Now this biological target, this term is actually frequently used in the pharmaceutical research to describe the native proteins in body whose activity is generally modified by a drug, and that results in a specific effect. And this effect may be a desirable therapeutic effect, or it may also be an unwanted adverse effect. Okay, so that is why finding a proper target is really essential. But sometimes the main protein due to which the disease is happening can't be your target. You have to sometimes look for another target rather than the main proteins which is involved in the disease. Why is that so? Because there are just like the characteristics of a drug, like there are some characteristics, we discussed the characteristics that a molecule must possess to become a efficient drug. Similarly, there are some characteristics that a biological target must possess, or a protein must possess to quantify or to what should I say, or to mark it as a desirable target. So first point that or the first criteria that it must have is that that particular protein must have a confirmatory role in the disease. It must be involved in the pathway of the disease directly it must be involved in there, or at least if it's not directly related to disease, it must alter your disease condition. Targeting that might alter your disease condition. This is the primary criteria. Okay, and the second thing is that the target that you are having, it must not be distributed throughout the body. Why? Because suppose again I'm talking about a lung cancer. Suppose you have lung cancer, or suppose you have this COVID-19. Okay, you have this coronavirus infection in your body. Now can you say how does a coronavirus gains entry into your lung cell?
From mouth.
Yes, from mouth. But what is the how does it gain entry into your lung cell? From mouth it is reaching your lung cell, but how is it getting entry inside the lungs?
By breathing process, I guess.
Yes, absolutely right. But I am much more concerned. I want to hear the name of the protein which helps to bring the coronavirus inside your body.
That I don't know.
Okay, the ACE2 receptor. Okay, the ACE2 receptor. There's a receptor present in your body, especially in the lung cell. The name is ACE2, and that ACE2 receptor is actually a receptor that is exploited by this coronavirus. So what happens is coronavirus goes and binds to this ACE2 receptor, and and then after binding to this ACE2 receptor, what happens? They form that particular coronavirus. The lung cell actually engulfs the coronavirus inside it. Okay, and as a result, it simply gains entry into the body. But the good fact is that this ACE2 receptor is not only present in lungs, it is also present in your liver and heart. It is also present in your liver and heart. So why coronavirus is gaining entry through the lungs ACE2 receptor? It's because the coronavirus gains entry, as you have mentioned, through breathing process. And when we breathe, the air only reaches to our lungs. It doesn't go to our doesn't move to our liver or heart cells. Had it had a infection through bloodline, then of course it would have bonded to your heart tissue. It can move into heart tissue or into your liver and kidney tissues. But since it is involved with the breathing process, it only reaches the lung cell. Now suppose I intend to design a drug. That can actually, so one way to prevent the entry of this coronavirus into your body is to shut down the ACE2 receptor. Suppose if I design a ligand and somehow the coronavirus fails to interact with the ACE2 receptor. If coronavirus fails to interact with ACE2 receptor, can it move inside your lung?
No, sir.
No, it can't move because it needs that access. Now suppose I have designed I have designed a drug that can go and bind to this particular ACE2 receptor. So what will happen if that drug is already bonded to ACE2 receptor of your lungs? Then coronavirus will have no place to bind there, right?
Yes, that will prevent it.
Yeah, that will prevent it. Simply like the musical chair. You have a lot of chairs, but if I am sitting on a chair, you can't sit on it. You have to find some other chairs, right? But
Yes.
but the problem is, suppose I design a drug against this particular ACE2 receptor. I am giving it as an injection or through your as an oral pill. So now since the molecule will flow through the blood, it will reach every tissues. It will reach every tissue. Thus it will reach the liver cell, it will reach the lung cell, as well as it will reach the heart cell. Now as I have mentioned, their heart and liver, they all possess the ACE2 receptor. So that means my drug will also go and bind to the ACE2 receptor of heart and liver. But this ACE2 receptor also has some other roles rather than binding to the coronavirus. It also has some other role. See, if I simply block the ACE2 receptor of some other tissues, I am preventing their actions, and as a result you might get a heart attack or your liver might fail to work properly. So in this way, the ACE2 receptor, though it is the most crucial protein, it can't be your drug target. It can't be your drug target if you are giving the drug in the form of an oral pill or through injection. Now, can you say, how can I bypass this complexity? How can I bypass this complexity of not allowing my drug to reach the heart or the liver cell? How can I prevent my drug? But suppose this drug is the revolutionary drug. This is the best drug that world has. So what to do in this? It's very simple. You only have to use it through your mouth as an inhaler. Just
So
like
that will also go to heart, I think, because
no. Just like inhaler. What happens when you are breathing? Is your oxygen reaching the heart? No, because because if it reaches your heart, then obviously the coronavirus will also attack your heart. But does it happen? No. When you breathe, the air goes directly to your lungs, not to any other tissues. So if I give that particular drug as an inhaler, if you have to inhale it, or I give it through a nasal drop, okay, if I give it as a nasal drop, or you have to breathe in that particular drug, then what will happen? All the drug will reach your lung cell, not to some other part or other organ of your body. Understood? So these are the things that you have to keep in mind. So even though your particular drug target is that particular drug target is present widely into your other tissues, but due to the role of the drug injection or the drug inhalation, I am preventing my drug to reach the other tissues, and I am focusing all my drug to move to the lungs tissues only. But if that particular case, that drug is not present in, suppose that particular thing is not present in lungs, or if it is a liver targeted drug, then of course, then what will happen? You can't use that. Then of course you can't use that, because then what will happen? That particular drug, if that particular receptor is present widely in your body, the drug will bind to every other tissues rather than binding to a specific tissue, and that will give rise to some other set of complication, some other unwanted effect. That is why whenever we need to find a drug target, we have to specifically see that this particular target is not distributed throughout the body. But yes, if it is a painkiller, if you are targeting a painkiller, or if you are using a generic drug like Calpol, paracetamol to bring the fever down, then of course you have to find a target that is present throughout the body. So it actually depends upon the context, the type of disease you have. But since specifically you are targeting about nowadays we only talk about some tissue specific diseases, then of course you have to have a target that can only be found in that particular tissue, not in some other tissues. Okay, understood?
yes, sir.
Fine. Third thing is that the target structure. You need a structure, because apart from the structure, it's really hard to design a perfect drug that will fit to your molecule. You can't see a complementary effect, right? That's why we need to have a target that have a proper structure. If you don't have a structure, then of course you have to design or solve the structure. Now, solving structure, there are generally 3 experiments that we use: either NMR spectroscopy, X-ray crystallography, or the cryo-EM structure. Now, of course, if you now suppose you have a drugs, if you have a protein structure that is solved by both X-ray crystallography and by NMR spectroscopy, then you have to select the structure solved by X-ray crystallography, because X-ray structures are much more better. They are much more authentic, because the NMR structure gives you a average value, average structure, but the X-ray structure gives you a much more detailed structure. But of course, sometimes when you don't have any structure, you have to go through the NMR. No, both NMR and X-ray are better, but of course, if you have a choice to select between two, obviously select the X-ray structure. But sometimes there are some proteins whose structures are not solved, because that protein structure is either very difficult to purify, or that particular protein can't. People have failed to crystallize that particular protein to solve its structure. Now, in those cases, either you have to look for other alternative, or you have to solve the structure by homology modeling. You have to use proteomics, you have to use structural bioinformatics plan. You have to use the structural bioinformatics to solve the structure, either by homology modeling or by ab initio. Now, ab initio modeling is not that it doesn't give rise to that better structure. So of course you have to move to the protein threading or to the homology modeling, and only by that you can design you can predict a structure of a particular protein, and after that you can design a ligand that can bind specifically to that protein. It is simply like identify in a spy movie. What happens? Suppose you hire a contract killer to kill someone. Now, of course, you have to give that contract killer a face, right? You have to give that particular person a face, that go and kill the person who looks like this. Suppose you are giving only a name. Go and kill Raj. Now there are thousands and thousands of Raj. How many Raj is he going to kill? Right? So that's why the drugs are also like that. You have to give it a face. You have to give the drug a face. Just ask him that go and bind to that particular structure, and it will go and bind to that particular structure only. So that's why structure is very much important. So if you find a particular protein that has a major role in that particular disease, but its structure is not solved, you can't use that protein as a drug target. You have to look for other alternative. It is because, and lastly, that you have is that particular target must be easily assayable. What is assayable? Do you know the meaning of this word assayable? No. Okay. So assayable means, suppose today I have I found a particular protein, and this protein is really the best target that I can have. Its structure is well solved. Okay, but that particular protein is difficult to purify. Now, as I have told you, that you have to find the target and you have to validate the data by doing the mol bio experiments, by performing wet lab experiments, by performing molecular biology, or by performing other cell biological experiments. But suppose that particular protein that you have, now the first thing that we have to find is the KD value, that with how much affinity that particular drug is binding to that particular protein. Now, can you tell me an experiment through which we can find out the affinity between the binding, either the drug protein or a protein, an experiment that gives you an idea about the KD value of your interaction? Okay, ITC, isothermal titration calorimetry. It's one of the method, or you can also perform fluorescence anisotropy to find out the KD value of their interaction. But the fact is that if that particular protein is difficult to purify, if that particular protein, but for finding out the affinity of course we have to have that purified protein. But if that particular protein is difficult to purify, or you are failing to purify that protein, then of course you can't perform any wet lab experiment using that protein. You cannot assay whether that particular drug is really binding to your protein or not. I can perform the bioinformatic assays. I can bioinformatical. I can say that yes, this particular molecule is binding to this atoms. It is using this interactions to bind to it. But you have to confirm and validate the data using the wet lab experiments. So until and unless you fail to find a particular purify that particular protein and perform all the experiments that you need to do, you can't use that protein as a target that is assayable. You have to do several biochemical assays. If it's an enzyme, then you have to perform the KM Vmax assay. You have to find out the after binding to your particular protein, is the Vmax coming down? What is the km of your enzyme before and after binding to your target, to your drug, that is the inhibitor? What is the Vmax of that particular interaction? We have to find this. If you fail to do this biochemical assays, then of course you can't use that molecule as a target, because you have to show those data. Without seeing those data, no government authority is going to give you a clean sheet of using that particular drug. You can't move to the next phase of clinical trials. You can't move to the clinical trial, and unless you perform the clinical trial, of course you can't market that drug, right? And finally, that particular drug must have a very promising the target must have a very promising toxicity profile. Now, what this promising toxicity profile means, that particular drug that you have, it must not be binding to it, must not show any other kind of toxicity in our body. For example, there are some proteins. Do you know what is the housekeeping genes? I think you should know this. What is housekeeping genes? No idea. Okay. So housekeeping genes means these are the genes which are required for your body's normal day-to-day function. These genes are required for your body's normal day-to-day function. Okay. So if you fall ill and one of this particular protein is involved, now to make sure, now suppose, how can I be sure that this particular protein is involved with my disease? How can I be sure that this particular protein is involved with a disease? Can you give me a experiment?
No, sir.
Oh, it's really easy. All you have to do is you have to knock out that particular gene. If you simply knock out that particular gene, what will happen? Knocking out of a gene means that particular gene, you are making that particular gene absent, right? You are simply deleting that particular gene. Now after deleting, now suppose that particular disease is happening due to the overexpression of that particular protein. Now if I particularly delete that gene, what will happen? There will be no protein in my body, and suddenly I am going to find that if I have no protein in my body, I am that particular mouse. We usually perform this in mouse. That particular mouse or the particular bacteria, they don't have that particular disease. They are not falling ill. That particular illness is removed from the body. Okay, this is the best thing to do. This is the phenotypic data, where there are some phenotypic changes that occur. Suppose you are blind or someone is blind. Okay, so you know that this blindness is coming from, it's an automatic blindness. Now this blindness is coming due to a particular protein interaction. You have a retinol, and your retinol is binding to some other proteins, and that is negating the activity of your retinol. Now I have identified a particular protein which is binding to the retinol and making it inactive. So if I delete that particular, and that is protein A, suppose. So if I delete the gene of protein A, there will be no protein A in my body to bind to retinol. And obviously, so in a mouse in which I am deleting that particular protein A, I am finding that particular mouse is able to see. Whereas in some other mouse where that particular protein A is present, that particular mouse is blind. So this gives me a phenotypic data that yes, that due to the role, this is the role of that particular protein. So since I am confirmed that yes, this protein A is actually inhibiting my eyesight, I can design a particular drug that can go and bind to protein A and rendering it inactive to bind to the retinol. Okay. But as I have said, that there are some genes whose function is required for your body's day-to-day activity. For example, that protein A is required for your ATP synthesis. It is a ATP synthase gene. Suppose it's a ATP synthase gene. So if I delete the ATP synthase gene, will that particular mouse survive? No, because without ATP we can't survive. We require ATP in our body. So there are few genes, there are few proteins. Inactivating them is only going to kill you. So these are the housekeeping genes. So if you delete the housekeeping genes, or if you simply design an inhibitor targeting the housekeeping gene, you are going to die automatically. Of course, when you die, you are going to find that you don't have that disease, but you are going to die. You are going to get cured of that disease, but you are going to die. So that's not a win-win situation, right? So that's why, even though a particular target, so your target must be, it must not fall under that housekeeping gene. It must, yeah, yes, sure.
So you said inactive that particular gene, that is protein A or another gene, we can cure the disease. So when we are inactive with the gene, the whole body is, when the gene present in our body, it is totally inactive in whole, or just particular in that eye area, or
No, you
where
have to
the
inactivate in the whole gene. Now, it actually depends upon the context also. Sometimes you know that your retinol is not only present in the eye, it is actually expressed in more or less in all the cells, in all the cells. And you can't perform this experiment. What happens when you do knockout? You have to simply have to take an embryo. You have to delete that particular gene in the embryo. So when the particular mouse is getting, so because when you get an adult mouse, all the cells of that particular mouse has that deleted portion. Okay, you have to perform this because you can't perform this experiment on eye gene, because in normal eye there are crores of cells, right? And it is impossible to perform this experiment in crores of cells. You can perform this experiment in one cell or two cell, but it is impossible to do in crores of cells. So even though if you manage to delete that particular protein A in one or two cell, it's not going to help you, because your eyesight doesn't depend upon the role of that particular one or two gene, no, one or two cell. It actually depends upon the thousands of cells that are present in your eye, right?
yes, sir.
That is why you have to perform this experiment in the embryo, itself. Whenever you have that, you have to whenever the particular sperm mixes with that ovum, you get a single cell. You have to perform that experiment on that single cell, so that when it divides and it becomes multiple cell, all the cells are clone of that particular deleted cell, so it will be the same. Understand? So that's why you can't use any gene which are a part of your housekeeping genes. For example, hexokinase. If you, as I have told you, that cancer cells are voracious eater, right? They have very high number of glycolysis, are very high, almost 100 to 1000 fold increase in glycolysis rate. So the best thing is to target the hexokinase gene. If I target the hexokinase gene, there will be no conversion of glucose to glucose 6-phosphate, and as a result the cancer cell is going to starve and die. But with the same thing, the same effect is also going to be found in my other healthy cells. My other healthy cells are also going to starve and die. So as a result, I am going to die. But the good thing is that I am going to get cured of that particular cancer. But nobody does like it, right? So that's why the particular target, it shouldn't be toxic to your body while deleting that. And the last thing is that it must have a favorable IP status. And this is not for you, this is relevant for the industries. And that is because suppose there are some proteins against which a particular drug is already designed by a particular protein. So if you need to design a drug against that particular protein, then you have to have that IP status, intellectual property status. And if you can't have those, then you can't market that drug. So these are some of the technical terms that are related to the patenting and marketing of the drugs, but that's not required for you. But you must also, if you are in an industry and if you need to design a drug for marketing, then of course you have to have this favorable IP tag on that particular protein that you are using. Okay, so now drug designing, there are actually two methods of designing a drug. Okay, first one is the reverse pharmacology, and another is the forward pharmacology. Now in forward pharmacology, what we do, it is actually a phenotypic drug screening. So phenotypic screening means I personally see that this particular drug, particular disease is happening due to this particular protein. So I'm going to identify that particular protein, and I'm going to see that yes, whether that particular protein is I'll read this first. So suppose I have a particular protein. So one way to design a drug is that you have a protein and you design a drug against that particular protein, which can bind to that exactly only that specifically to that particular protein using the particular groove or to its active site and whatnot. Okay, this is one method. And the other method is that you have a ligand, you have a drug, you have designed a particular molecule which has very good drug likeness. It can be used as a drug. Now you need to find a particular, now you have the structures of thousands of proteins and you dock your molecule to thousands of different proteins to find out the protein to which your molecule binds to. Now go back and try to find which disease is modulated by that protein. Okay, so these are the two approach. So in forward pharmacology, what we have, we try to find out the molecule and then just a minute, I think you can't see. Yes, we try to find out a molecule, then we identify a particular target after that. But in reverse pharmacology, what we do, we identify the target protein first and then we screen a compound that can bind to that particular protein specifically, and then we perform the other assays or the experiments that we need to do to mark it as a, or to brand it as a particular drug against that particular protein. So what do you think, which of the process is better?
So reverse pharmacology.
Reverse pharmacology is much better, obviously much better, but in industry the forward pharmacology approach is very much what should I say, it is also used, it's very much used because in the industries they have a huge database of ligands that have high drug likeness. So whenever a particular disease is coming in, suppose this, now we have this COVID-19, so in after 10 years we're going to have some other virus or other bacteria. So what will they do? They will simply use that set of whole library of drug against that particular viral protein. They're going to perform the proteomics, identify all the proteins present in that particular bacteria or virus, and then they're going to use that drug onto that. And if they find a particular target that yes, this ligand is binding to this particular protein with very high affinity, they are going to brand that particular target as a brand that particular protein as a target, and then they are to market it. But in academic industries and the people who are a little bit sensible, they prefer this reverse pharmacology approach, because in this case I can design a ligand that is specifically tuned to fit to that protein only, right? It is specifically tuned to bind to that protein only. So we have a much more what should I say, binding affinity. It will be much more selective. But nonetheless, both the method are very well known and well established. And one thing you will be surprised to know that there are some drugs in our market. There are some drugs in the market for which the target is not known. We don't know how that drug is actually recovering us, but we use that. Do you know the name of that particular drug?
So repeat the question.
As I've told you that there are some molecules or there are some drugs that simply bind, there are some drugs that actually bind to that particular protein, right? A drug must bind to a particular protein to show its activity, and we must know to which target is it binding. But there are some drug in the market for which we don't know what the target is, but since it is recovering us from a particular illness, we use that. We don't know the mode of action of that particular drug, but we use that since we are getting recovered. You know what? The first thing is that homeopathy. The whole homeopathy world is based upon this idea. They don't know what is happening, the mode of action, but they are using it. And the second thing is that widely used paracetamol or Calpol or Crocin that we take to suppress the fever. You can simply Google it out. You are going to find that this particular protein, this paracetamol, it doesn't have a target. Though people say it might bind to this protein A and protein, I'm forgetting the name of that protein to which it might bind, but they use the word might. They don't know for sure that that particular molecule or paracetamol is binding to that target to reduce the fever. But nonetheless, we use paracetamol because it is non-toxic and it is really capable of bringing down the fever, right? So both the methods are there, and there are some, as I've told you. So this drug world, you don't always have to find a target. If you can simply find a ligand that can suppress your whatever you are having, suppress that particular toxic thing that you in your body and suppress that illness, you can simply market it. No need of finding the mode of action and all. Okay. So that's why these are the two methods. In one case, you have a compound. You find the target, and this is the target deconvolution method. When you have a compound, you find the target, and you have your compound beforehand. So you are trying to find out the target to which it binds to. This is known as target deconvolution. And the other way around is when you have the reverse pharmacology approach. When you have your target and you want to find a compound that can bind to your target, and after their binding you have to go for the target validation and then the rest of the thing. Understood? So as I've told you, this target deconvolution is a thing that I've already discussed, right? When you don't have a target. So in this case, what I have to do, firstly, the thing that you have to do is, once you have your ligand, you have to screen the ligand to bind to all the proteins that are present. Now, do you know where all the proteins are present? There are some database which houses the information of all the protein structures that are present there, and you have to take the protein database. There are some protein database from which you can find all the information about a particular protein. So let's take a look into that particular database. Okay, I've sent you a link. Please click on the link. So this link is actually the PDB database. So what is this PDB database? This is a protein database. So this, as you can see, that this particular database has 174507 biological macromolecule structures enabling breakthrough in research activities. That means this particular database, if you solve a structure of any biological macromolecule, be it a protein, DNA or RNA, you submit that data into this database. This database has all the information about the solved structure of all the proteins whose structure is solved till date. For example, suppose I want to know about NCL. It's a nucleolin protein. Or suppose, as I've told you, let's talk about VEGF protein. Okay. So see, under VEGF, if I type in the name of the protein, you are going to get all the solved structure of that particular protein. For example, see the crystal structure of VR1, a VEGF from snake venom. This is also belongs to VEGF from snake venom. Oh, not the snake venom. I don't know why I'm not getting it. Let me find the NCL protein. Okay, so I have given the name hexokinase. So now if you see that under if you simply name it hexokinase, you're going to get all the solved structure of the particular hexokinase. So you have the hexokinase from this particular organism, you have hexokinase from the rat. Okay, you are going to have all the type of solved structure of the hexokinase. So let's take a look into this particular structure of the hexokinase. So you click it here, it's first is going to describe what this structure is: the rat brain hexokinase type 1 complex with glucose and inhibitor glucose 6-phosphate. Okay, obviously because the glucose the role of hexokinase is to convert glucose to glucose 6-phosphate. So in this case they have designed an inhibitor for glucose 6-phosphate, and they have solved the structure to show how the ligand binds to this particular hexokinase. Okay, now if you come down, it's going to see you the name of the organism, the name of the protein and the organism from which to which this particular protein belongs to, and the name of the author who have solved this structure. Okay, and they have solved the structure in 1998. You can see now this is the experimental data that you have. They have solved the structure by X-ray crystallography, and the resolution is 2.8. Now can you say what is resolution? Just think of the word resolution. Just give me a minute. Yes, what is resolution?
Detailed picture. I mean,
What?
level of detail, how the variation of atom in a structure.
Very good, very good. But in general, the resolution means that how far you can distinguish 2 points. That's very simple. So just make 2 dots on a single paper. Just make 2 dots one after another nearby, just side by side, and keep that paper in front of you and simply move away from that paper. And after a certain distance, you're going to find that particular, you can't distinguish those 2 dots as a single dot, right? They're going to feel like a single dot. It's simply like the train rail line. If you simply stand in front of a train track, what happens after a certain distance? You find that those train tracks actually merge together, right?
Yeah.
So that is the resolution. So in this case, 2.8 angstrom is the resolution. That means they can distinguish 2 atoms which are separated by 2.8 angstrom as 2 separate atoms. If those 2 atoms, if the distance between 2 atoms is 2.7 angstrom, they have taken them as a single atom. They can't distinguish 2 atoms if they are not placed, if they are not separated by 2.8 angstrom. Any atoms or anything which have a distance between less than 2.8 angstrom distance between them, they will take them as a single atom. So in any X-ray structure, if the resolution is below 3 angstrom, it's preferred to be good. But the lower the better. There are some structure in which you can find the resolution as in 1.5 angstrom resolution also. So the lower the value, the better. Okay. So these are the experimental parameters and setups. And if you simply come down, you are going to find this particular protein, and these are the things that are present in the protein. So this is the G6P, the glucose 6-phosphate inhibitor. Okay. It's a glucose 6-phosphate, and along with it, it has this glucose 6-phosphate, this beta D glucopyranose, and of course it has the calcium ion attached to this particular molecule. But now, how to visualize the structure? See, in this upper part, you have an option called the download files. Okay, so if you simply click that particular download files, you're going to find the option called PDB format. Okay, simply click that PDB format, and that particular file is going to get downloaded. Okay, so I'm going to talk about it later. So right now, if you simply move this screen, you're going to find the structure of this particular molecule. And if you need to visualize this particular molecule in your In the server only, simply click this 3D view. After, just beside this structure summary, you have an option called 3D view, and if you click it, it's going to show you the structure. It's going to show you this is the structure. And if you hover your pointer to wherever, if you simply scroll your mouse wheel up and down, you can zoom in, zoom out, and if you hold your left mouse button, and you can simply rotate the particular molecule to visualize its structure. So what I can see, these are the alpha helix strands, these are the alpha helix, and whereas these are the beta sheets. See, these are the beta sheet, these are the beta sheet, and see, and this is the whole structure, and this is the whole sequence of the whole sequence of your protein. This is the whole sequence of your protein. And to identify that which atom is which, suppose, see, as I am pointing to any atom, as you can see in the bottom of the screen, in the bottom, in this particular part, you can see that it can identify the atom, not the atom, it can identify the residue. In this case, it is the lysine of lysine 501, this is glutamine 502, this is arginine 500 arginine. So since this is a almost 800 amino acid long, the rat hexokinase is almost it is 918 amino acid long. If I'm not wrong, the rat hexokinase is 918 amino acid long. So if you hover your pointer and you can see the amino acid number and the position. Okay, understood. And these are the so see, this is the molecule. This BGC is the inhibitor. This is the glucose 6 inhibitor, the BG6. So as I can see that this BG6 is placed in this particular portion, that means it is interacting with this proline 605 and with it is nearer to this region, and this it is also interacting with this glycine 681 or with threonine 682. Okay, so that means these are the residue that this particular inhibitor is utilizing to bind to this particular hexokinase. And on the other hand, you have this molecule here, the glucose 6 phosphate, the glucose 6 phosphate. So that means this is the active site. Since glucose 6 phosphate is present here, that means this is the active site, because on the active site only the glucose molecule binds there, and it is converted to glucose 6 phosphate. So wherever this particular molecule is binding to, that must be the active site. So this is the active site of this particular protein. Okay, so this groove is the active site. But you can visualize it much better, but it just, this 3D screen only allows you to visualize before you download the particular molecule and to know. And there are other options like annotations, the experiment that they have followed, this molecule, which family they belong to, the sequence of the particular protein, and everything that you need about these things. Okay, so these are the sequence, and it also classifies the helix region, the beta strand region, the binding site. See, these are the binding site. So how do you know which is the binding site? See, as I point my here, just take a look into this area. See, it's saying a position 155. So that this on this position that molecule is binding to. And what is the position? What is the amino acid sequence? If you simply scroll your mouse wheel on this particular window, it is going to get enlarged, and you can see it is binding to this E. E stands for, what does E stands for in amino acid? E stands for glutamic acid, right? E stands for glutamic acid. So you can see that it actually binds to this particular molecule here. Understood? So with the help of this particular database, you can actually download a particular protein structure. You can download any protein structure that you want. Nothing that you want. If that particular protein structure is solved, it will be present here. Or you can also find the structure of some DNA. For example, 1XAV. 1XAV is the PDB code. Like every protein or every DNA, they have given a PDB code. So this is the G quadruplex structure that is present in the c-Myc promoter. So why do I know this? Because I have designed a ligand, I have designed a drug that can specifically bind to this particular G quadruplex structure and stabilize it, and thereby reducing the c-Myc protein. And c-Myc protein is actually found to be overexpressed in almost 90% of cancer cells. And till date, nobody has found a drug against c-Myc protein or c-Myc gene. So that's the first time I have developed a drug against this particular protein. So that means this is also a G quadruplex or a DNA structure. So you can also find, if any structure is solved, you are going to find them in this particular PDB database. Okay. Now I'm going to use this particular, how to use the protein, how to use this PDB site to understand various things. I'm going to discuss that in the next class. And for that, I have given you a software, right? PyMol. Please do install that PyMol into your computer and keep it ready if you haven't done already. Then because we are going to need the help of PyMol in the next class to analyze a particular protein structure, to find out the nitty-gritty details about the protein structure, how to identify the binding site. We're going to perform those things. Okay, so now let's get back to the particular session. And so now we know what is target discovery and what is target deconvolution, right? So we know this. We have idea about what is what. So one belongs to the reverse phase, reverse pharmacology, and target discovery belongs to the target deconvolution belongs to the forward pharmacology, whereas target discovery belongs to the reverse pharmacology, and we have idea about what is what. And as I've told you that this target discovery is also very important, because only when you have a protein, if you have a protein, then I know that what is the target and what is the binding site. And without knowing the binding site, if I know the binding site, I can exploit that particular binding site to design a ligand that will bind to it much more specifically, right? So that are some advantages that this particular phenotypic drug discovery gives you, and that helps you to design a particular molecule, and that can actually be much more specific. Because I'm repeating the word specific because this is where the drug industry actually fails to design a specific drug. And since more or less there are several proteins which are homologs to one another, and since the drugs are really small, as you can see that suppose, see, in this particular case, this is the inhibitor, right? This is the inhibitor. So since this is the inhibitor, now see how small is this, and these are the interaction. If you simply click that particular inhibitor, it's going to show the interaction, that it is forming with all the other nearby molecules. What are the interaction it forms to, how does it bind to. So see how small is it. It's a really small molecule. Now it can easily fit into anywhere. It can easily fit into any protein, right? So that's why if you are giving this particular inhibitor into your body, of course, rather than binding to hexokinase, it can bind to any other protein. It can bind to any other protein, and if it binds to any other protein and starts to inactivate that protein, then of course that's not my intended target, and it will give rise to some serious side effects. Understood? It is going to give rise to serious side effects. So that's why specificity and toxicity, these are the two most things that are highly required for any drug. The specificity and the toxicity. So I'm going to repeat this specificity several more times, because unless your molecule is specific, you can't do anything. The foremost thing that your molecule must have is the non-toxic nature, and second thing is the specificity. The more specific your molecule is, the more better your molecule is. Okay, the more price you are going to get. For example, the lab where I worked, we have already discovered a anti-metastatic drug, and we are in now talk of marketing that particular drug to a company. They are going to perform the clinical trial. But since that particular molecule is really selective to a particular RNA, and the price that the company is going to offer is 100 crore. Obviously, among that 60% is going to be taken up by the institute where we have performed the work, but the rest of the 40%, that means almost 40 crores are going to come to that particular professor who have discovered it. So just think of the huge money that they are giving. And in their case, this 100 crore is really less. 100 crore is really less, because sometimes the drug price can go up to 10,000 crore or 20,000 crore. The particular person is going to, the drug industry is going to pay you to design a particular drug. So you know that this is a very, very great set, and where you can thrive. And if you simply need to make money, if you have the idea of making money, then if you can design a drug, a very specific drug, then your life is set. Okay. Okay, so yeah, so now as I've told you that after we have found a target, we have to validate it, right? We have to validate that yes, this is the target that I'm talking about. This target can actually reduce my what should I say? It can reduce this particular pathophysiology. It can reduce the illness. This, but if targeting this drug, but I have to make sure that this target is actually plays a key role in this particular disease. So how to perform those? There are various techniques for validation. Of course, you can do the disease association. You can design bioactive molecule. There are cell-based methods to do so. And for example, as I've talked about, first thing that you can do is obviously do the bioinformatics. You have to perform the bioinformatics to identify, select the particular target, and then you have to see that whether that particular target expression changes according to the nature of the disease. As when the disease progresses, that expression is either increasing or decreasing. If that particular disease is due to the overexpression of a protein, so as the disease progresses, the expression level will increase. And if I'm treating my drug to that particular protein, so that particular protein expression will decrease, and with the decrease of protein expression, the disease is also getting eliminated from my body. So these are the expression profile measurement that you have to do, and you can perform this with simply by RT-PCR, or you can simply perform the PCR experiment to know that if it is decreasing or not. And finally, you have to perform this functional screening by developing the knockout assays, or by simply designing an RNAi that is going to bind to that particular protein or the particular portion of that mRNA and prevent the formation of protein. And if you don't have that protein, that is a good sign. That means that you don't have the knockout gene, right? If I am preventing my protein translation, then that equals to the knockout sector, right? If I knock down, means I don't have that particular gene, so there is no protein. So rather than creating knockout, I can also use RNAi to interfere the protein translation. And if I don't have any protein in my body, that is equal to having the knockout, right? So these are the experiments that you have to really perform to check that, validate that yes, the target that you have selected is really your target, is really the target. And modulating the expression of the target can really that can hamper either the protein, the progression of that illness, right? So you have to make sure those. So these are the things that you can do. Okay, so that's all. So that's all for today. Okay, and in the next class I'm going to talk about the other thing, like I want to show the PyMol, how to explore the protein target, or how to explore the protein PDB molecule to find out the different avenues to measure the distance, to find out the which atoms are involved in the interaction and all. Okay. Okay, fine. So thank you for joining this class today, and I'm going to send you the next class will be on I think it will be on Saturday. Okay, I'm going to mail you the schedule. All right. All right. Thank you.
Thank you.
Thank you.